Welcome to Proteins and Enzymes Part 10. In this tutorial we are going to look at enzyme classifications and begin recognizing the types of reactions each different classification will catalyze. Let's get started. Alright, so we're going to focus on 6 main classes of enzymes. You know, there's always more when people get into details, but this is the heart of the matter. Alrighty. So as in our study of functional group reactions, we see that oxidation and reduction are very important. So there's no surprise here. The oxidoreductase enzymes will be easy to spot because you'll recognize an oxidation and reduction reaction, and they have a huge flag. They always write, because it's redox, we need, don't seem to be able to write, there we go. So they write they require a coenzyme. So there's always a very obvious flag for the oxidoreductase enzymes. We'll see NAD+ with NADH, or FAD with FADH2, or NADPH with NADP. So look for those coenzymes. It's a big, huge flag. We will also see that transferase, right? Transferring functional groups. So very frequently we will see the phosphate group transferred, and often symbolized with like a P1 or a PI. Acyl groups also very frequently transferred, or amines. Often you will see kinase in the enzyme name. And so as I mentioned these functional groups, ideally in your mind you see them, right? So if we think about a phosphate group getting transferred, may just be straight away like that. Acyl groups, right? Think about that carbonyl. Here would be the acyl group, or amines. Alrighty. Now, we have water will show up in a couple of these. The first important one is hydrolase, right? And so this is hydrolysis. This is the splitting apart. So when you see hydrolysis, you can think about it being, right, catabolic, right? And not only that, we can think about it being exothermic. So we can bring in all of the concepts we've been learning about reactivity. And so in this case, right, we will have a big, large molecule, and then water will get added. And when we're thinking about hydrolysis reactions, it's really helpful to think about water as HOH. And then we know that the hydroxyl group of the water will bond to a part of the molecule, and the hydrogen will come in and bond to the other part. And so we could look here and recognize that, right, our large molecule is going to be split into pieces, and we get 2 smaller molecules. And the other thing to note is that the water, right, part of the water goes with one molecule and part of the water goes with another molecule. Now we will see water in this next reaction, and so it's important when we see water, we can't go on autopilot and just think it's hydrolase. We have to look at the changes that are occurring to make the distinction between a hydrolase, and in many cases a lyase will involve water. So let's look at lyase next so we can compare and contrast. The big deal with the lyases, to keep in mind that it's all about carbon-carbon or carbon-oxygen double bonds. So that's the flag there. So unlike the coenzymes for oxidoreductase, the double bonds can be our flag to help us recognize what type of reaction is occurring. So here we'll just use a carbon-carbon double bond, and who knows what's going on with the rest of the molecule, right? And then here comes our water, just like before. However, now we notice, right? So the water is going to merge completely with this molecule, right? So here's our carbon skeleton. And now the hydroxyl group will go with the carbon on one of the carbons, and the hydrogen goes with the other carbon. So here we've taken, right, we've taken two molecules, and we've made, it's one molecule. So in this case, so I think that's enough to say there. And so it'd just be important to compare and contrast. Just because we have water, we have to look and see. So I've given you a couple flags to help you distinguish. And now we'll look at isomerase, and very literal, right? It's going to be an isomerization. And so we will have a compound. Biological molecules tend to get very big, so at a minimum, we will have carbon, hydrogen, oxygen, nitrogen. We may possibly have some phosphorus as well. And then when we look at the product, we will see that we have the exact same number of carbons and hydrogens, oxygens, nitrogens, right? So you might think, well, nothing changed. But if we look more closely, we'll see that this has different connections, right? Different bonding, right? Different connections or spatial arrangements. All right, so when we think about isomerization, we think about the types of isomers that we've learned about, right? So they could either be structural or they could be stereoisomers. So a long time ago, we talked about, and we were talking about double bonds. We talked about, like, while in the active viewing of this tutorial, we have cis double bonds and trans double bonds interconverting to trigger nerve impulses. So that would be where we might have an isomerase enzyme catalyzing the reaction. And in our biochemical pathways, we very frequently see more structural isomers. We change the connections of the atoms. The last type of isomer that we're going to look, or excuse me, enzyme we're going to look at is the ligase. Ligase is French for ligaire, which means to link together. So a bond formation. So we have bond formation, and it's coupled with ATP. So this is the kind of the opposite of hydrolysis. We could think of this as then as anabolic or endothermic, right? And it's endothermic, right? Because the reaction to be catalyzed is endothermic, we couple it with ATP as the energy source, right? So the ATP will be the E source. All right, so symbolically, what does this look like? So it's going to be basically the opposite of hydrolase. So we have two molecules, and typically one of them will have a hydroxyl group, and the other one will have an ionizable hydrogen. And so we're going to link these two, couple these two molecules together. But because it's endothermic, it won't happen on its own. So we bring in some ATP, and it will hydrolyze to ADP, and in the process release energy, which couples to this reaction so that we can produce a larger molecule, and then we have water as a byproduct. So we can see that we took two smaller molecules, and we produced a larger molecule with water. So when you get to your bio courses, it's really common to call this, right, synthetic dehydration. right? So there's more than one flavor of dehydration. So this is synthetic dehydration because of the synthesis, the two small molecules into the larger molecule. Alrighty, so there is the big picture and introduction to these 6 main classes of enzymes. Now let's go back and look at each one a little more closely and with some actual biochemical reactions. So we'll start with oxidoreductase, right? And these will be the redox reactions. And so we start to look, and here's our flag. Oh, look, we see a coenzyme, and we look at our substrate, and we notice we have to look for the change. So we notice a carboxylate and a methyl group. Those are all the same. It's pretty obvious, right? We have our alcohol group and a ketone group. So there's our change. So let's go ahead and review. So what happened here, right? We had an increase in our carbon-oxygen bonds. So this is definitely an oxidation. And then if we look at our coenzyme, we notice, oh, there's no hydrogens there, and there's hydrogens there. So we would recognize this as a reduction. And then the lactate dehydrogenase, right, we recognize that as our enzyme. So we understand every role. All right, and so basically we've, see above, we have identified the oxidation, right? So what is this? So this is lactic acid, right? So lactic acid is getting oxidized to pyruvate. And let's see, what else do we want to do here? Lactase dehydrogenase is the enzyme. What is the name of the term given to NAD+, right? So that's right up there. It's the coenzyme. So that's our flag. So there's several ways to recognize an oxidation-reduction reaction. Just looking at the substrate for its change, and then the role of the coenzyme, because oxidation-reduction reactions always happen together. Alrighty, now let's look at a transferase reaction, right? So we're not going to look at transferring of all the different groups. We'll focus for this example on a phosphate group. So if we look here, we see we have a glucose molecule, often just gets symbolized with a G. And if we look here, now we see we have glucose 6-phosphate. So we look for the change, and we see here we had an H, and now we have the phosphate group. So we've transferred a phosphate group. Where did the phosphate group come from, right? It came from the ATP. So we have to be careful with ATP as well. Notice that that in this case the ATP is the phosphorus source. All right. So we've transferred a phosphorus from ATP to the glucose. So the hexokinase is a transferase enzyme, and let's draw the structure of the functional group transferred. So let's keep in mind what phosphate groups look like, and we'll go ahead and do it at physiological pH, and we'll leave one of these as an alcohol because we can look here, right? And so this is our, basically glucose is a very fancy alcohol, and here's our phosphate. So it's phosphate ester formation, and so and so the hydrogen of the alcohol and the OH group, right? So we would also produce water here. Alrighty. Now we will look at ligase. Remembering ligare, bond formation to link together. The flag here is we'll always have an ATP ADP reaction. So here we have pyruvate, and it is getting linked with carbon dioxide. So when we and then we see our final product here. Notice that our final product does not have any phosphate added to it. So when we look at ATP here, we see it as an energy source. There are several ways this could be written. We might also see the ATP here with the ADP to show that they're a coupled reaction. And we think, let's see, what else can we think about, right? So here we would say this is A, right, and this is B, and now we see that A and B are locked together. I guess more accurately would be B over here, and then here's A. So things have been rearranged a little bit, right? But we see that we have linked two small molecules together to make a large molecule. Right? So we've taken two small molecules, we've linked them together to make a large molecule, and we needed ATP as an energy source because we don't see the phosphate molecule in our final product. Right? So now it would be a good moment for you to reflect on a way that makes sense to you. When we're comparing transferase to ligase, what clues are you going to use to distinguish between them? So for me, what I look at, right, is that the ATP, right, is the energy source, right? No phosphate on final product, right? And then we have two smalls to one large. For transferase, for me, right, the phosphate, right, appears bonded to the product. And if something else makes more sense to you, then that's what you should write, because these notes are to help you. Alrighty. So let's go through a couple more of the enzymes in greater detail. Let's start looking at hydrolase a little more closely. So lipase is a great enzyme for catalyzing the hydrolysis of fat. So let's look here. So we would call the fat or the oil, the triglyceride, the substrate. And then, of course, we have water, and I think it's helpful to continue looking at water as a hydroxyl group and a OH. And then notice that we end up with 4 smaller molecules. Right, we'll have 1 glycerin and 3 fatty acids. Alrighty. So let's draw a line through the bonds in the reactant or substrate that are split apart by these water molecules, right? So where in the fat does this water come in to do hydrolysis, right? So remember, our carbonyls is where we can get our bearings, and we know that the water always comes in between the carbonyl and the heteroatom. So this is where the water is going to come in. It's going to give an H to the alcohols, and the carbonyl always gets the OH. All right, so we've drawn in the line to show where the bonds are broken. Now, let's look at the products. Let's circle the hydroxyl groups in the product that were derived from the water molecules, right? So that would be right here. So from the water, there are the hydroxyl groups that came, because we know that the hydroxyl groups always connect with the carbonyl carbon. And now let's box the hydrogen atoms that came from the water. And so those would be right here. We'll just, there we go. Okay. So, all right, so that's about it there, right? So we've had a lot of experience with hydrolysis reactions. So we recognize the ester bonds, hydrolase, to carboxylic acids and alcohols. Alrighty. So now let's look at Lipase, another enzyme that frequently has water as a reactant. So we'll look here at carbonic anhydrase. So we have carbon dioxide and water, right? So with liases, they catalyze the addition of groups. Water is very frequent. We could see CO2 or ammonia, right? Remember, the key with liase is it's a double bond. And remember that when we talk about enzymes, they can be forward or reverse. So we could either add to a double bond, or in the reverse reaction, we could form a double bond. So remember to think about things both ways. So here, let's move the water over to make it a little easier to see. And let's look at so here's our initial reactants and here's our products. So this one could seem a little crazy, but well, here's our carbon, right? So let's look at what happens here. And what happens is, right, we see that our carbon is going to have a new hydroxyl group. So this is going to come here. And we notice that now there's only one carbon to oxygen double bond. One of the carbon to oxygen double bonds becomes a hydroxyl group. So the enzyme creates bond formation where we've shown with the red arrows. And then that leads us to our final product. So we can see here that we have added water, right, to a carbon to oxygen double bond, all right? And it could also be carbon-carbon double bonds, but this one is a good example. So let's keep that in mind up here, right? So the double bond could be carbon-carbon or carbon-oxygen. Alrighty. And so then we can keep track, right? This was from water. So notice once again, right, that we had two molecules, right? One of those molecules was water, and we ended up with one larger molecule. So we've used that description a couple times. However, notice though that here water is one of the molecules that's added across, and we have the feature of the double bonds, right? So I've noticed in the past that it's frequent for students to have a little bit of trouble distinguishing between hydrolase and liase. So now's the time for you to write some comments to yourself that help you distinguish between these two enzymes. And I'll write mine, what helps me here, to guide you, right? So for me, the whole point of hydrolase, right, we can think about that it's catabolic, right? It's really the opposite, right? Water splits apart two molecules, or splits apart a molecule. Right, into two smaller molecules. All right, versus liase, right? So what's going on with liase? So with liase, basically, right, it's the water bonds to the substrate. And where does it bond to the substrate, right? At a carbon-carbon double or a carbon to oxygen double bond. All right? So, okay, so that's the main ways to distinguish between hydrolase and liase. So now we'll look at the last classification, of enzymes, the isomerase, and then we'll just practice identifying and classifying enzymes in a variety of reactions. Alrighty. Okay. So the last one here, we have two molecules, and we look at them, and as we start to scan them, it's like, I see a carboxylate, I see a hydroxyl, I see a phosphate. Often you can go, I'm not seeing any differences. There's the minus charge, right? So I'm not really seeing any differences. So when you feel stuck, like, I don't see anything different, do a quick inventory, right? We'll see that we have 3 hydrogens, excuse me, 3 carbons, 4 hydrogens, 7 oxygens, and a phosphorus with a net charge of -3. If we start counting atoms on the right, let's see, 1, 2, 3. All right, so C3, H4, O7, phosphorus 3-. So now we see what happened, right? We have the same chemical formula, but we have different connections. So the relationship between the substrate and the product would be that they are structural isomers, right? Making sure everybody feels comfortable with the language, right? Substrate and product. And so in this case, it's not right? So if it was geometric, right, that would be a spatial arrangement difference. But here we have different connections. So they're constitutional or structural isomers. Okay. So we've gone through the 6 major classifications, and they're written out for you right here. And now we have 6 reactions to look at. So I can just we'll just have to, there we go. We can't quite get them all. All right. So now we want to practice. So this would be a good time to pause the video and practice classifying them on your own. And then you can compare your answers with mine. Let's look at this first one. It can be very helpful to circle where we see a change. So we look from left to right and we're like, there's a double bond here and there's no double bond here. But I do see the addition of water, right? So we have 2 molecules forming a larger molecule and with, a double bond, so lyase. So we would need a lyase enzyme to catalyze this reaction. Now we see another example with water present. This molecule is urea, and we notice that urea reacts with water, and now we have 3 molecules, right? So what has happened? So we see hydrolysis happening, and we would actually, this isn't balanced, we would need to add some more water here. I think we would need 2 waters, right? So the water is coming in and it is splitting apart the urea. So we've gone from a large molecule, right, to smaller molecules. And remember, everything's relative. I know urea doesn't look that big compared to some things we've been studying, but if we compare it substrate to products, it's the biggest molecule in this reaction. So this is definitely an example of a hydrolase. Okay, we'll go to our next example. We look here and we say, okay, there's an alcohol. Ooh, did it get oxidized? Let's keep looking. But then we see a carbonyl and an alcohol. So this doesn't appear to be oxidation and reduction because our substrate has a hydroxyl and a carbonyl, and our product has a hydroxyl and a carbonyl, and then phosphate groups. So there's no oxidation and reduction, and there's no coenzymes. So often when I'm stuck, then we go, oh yes, isomerase. Right? Nothing has changed. We've simply rearranged the parts. Okay. Let's look at reaction D. Here we have pyruvate and CO2. Oh yeah, and this one formatted a little differently. We can see this part's all the same here. And then here's our CO2, and we can see the carboxylate. All right, so we have two small molecules. We've linked them together to make a large molecule. The phosphate, nowhere to be seen, so we recognize this as an energy source. And this would be, so pyruvate carboxylase is a ligase enzyme, ligare, linking together. Let's look at reaction E. We start here, we have a 2-carbon chain with an alcohol. Whoa, there's our coenzyme. We pretty much already know what this is going to be, but let's follow up, right? We go from an alcohol to an aldehyde. So we have oxidation and we have reduction. So this would be an example of the oxidoreductase. And then our last example here, we look and we see we have our buddy glucose, and we look over here and we go, ah, there's no, so that's not glucose 6-phosphate, right? But there is a glucose phosphate there, glucose 1-phosphate. So we've added a phosphate group to glucose. So here the ATP is not an energy source. I mean, there's still some energy shared, but really we're focusing that it's the phosphate source. And so we would classify this enzyme as a transferase. Alrighty, okay. Well, that concludes our tutorial on classifying enzymes. Please take some time now to work a few homework problems to reinforce your understanding.